everybody, it's Mr. Chemist for AP Biology. So it's nice to see you and hope you're enjoying your snow day. We got 9 or 10 inches on our back deck this last night, so it's probably good we're staying home today. But we do have some work to do. So I wanted to check in with you a little bit, give you a little bit of a lesson, and to kind of further point you in the right direction for what to accomplish. Let's go to the share screen. Okay, so for sharing, this one we did in class on Tuesday, but it was a little hard to follow along once they announced this. So you need to finish that up, like go over some of the answers to make sure. And then there's 3 things for you to do today and tomorrow. The timeline of life on Earth, life starting questions, of life. So I'm gonna go through those different parts of the lesson at home learning today. So all of this information can be found on the schedule. So we kind of organized this before we left on Tuesday. We have these 3 assignments here. Also keep up on your reading in chapter 24 and 25. Those ARGs are really helpful at summarizing and guiding you through the different work on the So I'm going to go back to the speciation concept. Some of these were kind of tough, and I did see some students get a few wrong at first, and then they were able to move things around, and you can find where stuff goes once you read a little bit further. But it was a lot of information, and nice to have it all organized. But it is someone else's thinking too, so it's kind of a challenge to look at and figure out where that would be. We start in the middle here. We have speciation. It's the formation of a new species. That one requires reproductive isolation. And so there's kind of a couple ways that we can organize that. You could have it pre and post-zygotic barriers. Remember, a zygote is the joining of the sperm and egg. Before those are joined would be pre-zygotic barriers. Then after they're joined, it's kind of after the organism is born as well. They're not the healthiest, or they're not fertile. And then we also talked about geographic isolation, like the sympatric versus allopatric. So if they're in different areas that allopatric speciation can occur, that's a pretty big idea. Separated by geography. And then if they're living in the same area, okay, we can have sympatric speciation. So that is usually here with the habitat or ecological isolation. They live in the same area, but they perform different roles, different niche. They can eat different things. They can live in different parts of a tree, like the top or the bottoms, or otherwise separate and not competing with each other. So we call them different niches. So the 2 examples for that was the garter snakes. One lives on land, one in the water. They're technically still in the same environment. And then the stickleback fish, if you watch that video, it kind of went through that one. Also the frogs and toads. They involve separate species after being separated into fresh and salt. And if we go down here too, it fills in some of the examples from this example sheet, example choices. And on this sheet, it's kind of nice. It's the same examples from the notes that we did in class, but it also gives a longer discussion with words of what is happening. So read through that, make sure you understand what is happening with each example. Then it'll make more sense. So, and then these words are pretty tough. Temporal, remember that's reading at different times. Example we talked about was silks. Behavior isolation, this usually is a bird call. But there's a couple other examples too: courtship rituals, dancing, mating displays, feathers, and even fireflies, different blink patterns. And this example with the seals as well. Different mating rituals. Mechanical isolation. These have morphological differences that prevent successful mating. So these are like physical body differences. They can't physically meet. So they had an example of bush babies there, which are relatively lucky. And the gametic isolation, this is where the sperm and the egg, they're just chemically different. There's lots of markers on the outside of cells that identifies it as a species. When they're different, they just don't mix. And so we call it gametic isolation. We saw that with plant pollens and sea urchins, among many, many others. Okay. All right, let's move over to the right-hand side. This is after the sperm and egg have joined. It's usually the resulting organism is not always true, but it could be different enough. They're not able to breed offspring, or they're not healthy enough, or even after several generations, they're just not healthy. They're physically deformed. They don't survive very well. But we kind of separate these into three. Reduced hybrid viability. This means they're not the healthiest, or there's some physical abnormalities that we saw. They don't survive well, and they don't fully form. Reduced hybrid fertility. This means they can't, once you breed these two organisms together, they can't have babies on their own. We talked about this with the mule. That's a full mixture of the donkey and the horse. You can get a mule, and it's very healthy. It's a great animal, very useful. Problem is, you can't take two mules and make a baby mule. The DNA is too different. They have one less chromosome, or one more, depending on who you compare it to. So you have to go back to the original horse and donkey. We call that reduced hybrid. Remember, a hybrid is a mixture of two different species. And then hybrid breakdown is kind of both of these two, and it's over several generations. So maybe the first one is healthy and can kind of have babies, but the second, third, fourth generations, it becomes less viable. So we see that in this example. You have to go back to the species that you're mixing to keep them healthy and vigorous or fertile. Hopefully that's useful for you, be able to see some of that. All right, how life started on Earth. This is really good background information, and it kind of talks about how life evolved. And make sure you hit some of the key events there. Earth started 4.5 billion years ago. Life started 3.5 billion years ago in the shallow seas near Australia and some very old bacteria. And there's quite a few conditions for that to happen. And we had to be able to have a cell. We had to be able to have some nucleic acids. We had to have proteins and getting energy. And so all these early conditions, we could try to replicate. And so in the 1950s, there's this very famous experiment by Miller and Urey. Make sure you know all the details of that. Take good notes on the Miller and Urey. That is one of your learning objectives, and there's a lot of details. So the mixture of gases, what was present, what wasn't? There's definitely no oxygen. How did electricity? Mostly enlightening there. And these organic compounds, what molecules can kind of self form and self-replicate, and were around during these pre-life years. Let the creation. Now, a key understanding here is that we can replicate the for life to occur, but we can't actually have life start from nothing in the lab. we're trying. We think we can do it. We've been trying for hundreds of years, even back to Frankenstein. And there's a couple of molecular geneticists in California that they're pretty close. They can get a cell, they can get the cell to divide, but they can't get it all together to make life a thing. But folks, would that be cool to work in that? Love it. I would, if some of you would end up make life with nothing. That would be so. Anyway, here's the first life on Earth, the stromatolites of really ancient bacteria. And then the key learning too there is there's no oxygen in the early atmosphere. There's no oxygen until we got to, which was about 2 billion years ago. And that rusted out. We kind of go through some of the other things here. Yeah. So that's a pretty good video there, how life started. The second one is a little bit shorter. You can look at chapter 25 in the book there, 25.1. It talks about the 4 key ingredients for life to start. Cells, those satellites, ancient bacteria. There's a little video that kind of talks about those different steps of the theory for life starting. And then here you could take really good notes on the Miller-Urey experiment and the role. For the theory is that RNA started single-stranded. It was a temporary copy of some sort of genes, and it was easier to copy only one. Then DNA somehow evolved later. It was double-stranded proteins. It was a more permanent copy. So that's how the theory goes. Take a look at that one. Kind of nail down those. And then the last assignment here, the timeline of life on Earth. Oh, 4.5 billion years ago, we had Earth forming. 3.5 billion years ago, we had life starting. 2 billion years ago, we had the first eukaryotes. 7, we had the first photosynthesis. 1.5 billion years ago. And then 1.5 billion years ago, we had the beginning of kind of life as we know it on Earth, called the Cambrian explosion. So 2 things happened there that were really important. Number 1, the evolution of hard bodies, okay, so shells. Being able to get away from that predator. And then because there were shells, we can also see the animals preserved much more thoroughly in the fossil. So that Cambrian explosion is really important. The Cambrian era is the name of that geologic period. Second thing that happened in the Cambrian explosion is because of those hard bodies, we started to see many new branches, many new forms of life. All of the major animal phylums or groups actually originated in this, which is pretty cool. Within a few million years or 100 million years or 50 million years, all of the organisms that we see today, ancestors, at this time, this Cambrian explosion. It's pretty cool. So those 2 things, hard bodies and all the major groups of really, we can see them in the fossil record because they had the hard bodies. A little bit later, we had colonization of land. Organisms left the water, fungus, plants, animals. And then the dinosaur death about 60 million years ago, that giant asteroid, Yucatan Peninsula in Mexico, led to the dinosaur extinction and the rise of mammals, the dominant life form on Earth. Just put those on the timeline here. Read through this different part, make sure you understand the history of Earth. This is a very brief history of Earth, but it's some of the points that you'll need to be successful. Right, that is the end of your lesson. After we get through that stuff, we'll we'll talk about the tree of life and planograms, the next chapter. And I hope you're having a great day, and we'll see you. I'm signing out.